Last time we were talking about grants. So we talked about sort of the mechanics for how they're reviewed at NSF, NIH, and Sigma Xi. Today what I wanted to do is I wanted to actually right now here on the board, write a Sigma Xi grant proposal. I'm going to actually let you guys pick what the topic of the proposal will actually be. And we'll do it in under 30 minutes just to demonstrate how straightforward it is to do. The reason I'm doing it isn't just for Sigma Xi. Again, I encourage you all to go ahead and do a Sigma Xi grant proposal. Those are due again October 15th. But the reason isn't just for that. The reason we're doing this is because basically the same procedure I'm showing you applies to any grant. The same thing applies to NIH grant proposals. I couldn't write the board in 30 minutes, but NIH grant proposals, NSF grant proposals, your graduate research fellowship, for those of you applying for that, the doctoral dissertation improvement grants, for those of you who end up applying for that in a few years. The same procedure applies. The same basically 4-paragraph formula I showed you guys applies, except that then it wouldn't be 4 paragraphs, it would be 4 sections. Just expanded out and that sort of stuff. Okay. Are there any questions before we go into that about some of the mechanics that we talked about last time for the review process? No questions at all? Okay. Now, so for Sigma Xi, let me just remind you of the mechanics for that one. This is a case where you're writing a 500-word proposal max. It could be less than that. It could be 490. 500-word proposal max. You can get 2 of them in the time you're in graduate school. And it's going to be reviewed by people who, to say they're non-experts is the biggest understatement ever. You'll be lucky if they're in the same general field. If you're a geneticist, you'll be lucky if it's read by a geneticist. And it may not be. It may be read by an ecologist. It may be read by a physiologist. It'll probably be, most likely, it will be a biologist, but there's actually a slim chance it wouldn't even be that. Okay, so it's a good challenge for you. All right, topic. Somebody pick. Wide open. Somebody pick.
Do you mean like a question?
Yes. Like what is this grant going to be about that we're going to write here on the board?
Adaptation to serpentine soils.
Adaptation to serpentine soils. That'll be our title. You have to forgive me because I don't even know what serpentine soils are. What is that?
They're like soils that are, I don't really know either.
This is going to be a hard grant.
I mean, it's like,
they're like low nutrient levels.
Low nutrient, okay.
And mineral content.
Okay. Okay, so this is the general topic. Thank you for that. I was like, I have no idea what that means. It doesn't have serpents, so. What's the question we're trying to ask in that? So the topic is adaptation to serpentine soils. What's the question we're trying to ask? Like the genetic basis of adaptation to serpentine soils, or, you know, I'm just tossing out things. I don't know. I mean,
how it affects speciation, maybe?
How it affects speciation. Ah, okay. How affects speciation. Okay. Just getting a couple of parameters up here so we know before we write the grant proposal, we have some idea where we're headed. So I'm assuming these are, since this is a soil thing, I'm assuming this is a plant proposal, right?
Yeah.
Okay. What's the system? Mimulus.
Mimulus, yeah.
Sure. All right. So mimulus, Gutatus. Let's say it's within Mimulus. Let's say within or among populations of Mimulus Gutatus. Okay. Okay. Let's see. So how it affects speciation, and particularly. I'm trying to make up something to go here to fill in the gaps before we actually write the proposal itself. Actually, let's start with this. I think this is good enough. I think this is all we need. Now, the criteria for all grant proposals, especially heavy on these two, this one as much as it relates to the others, and these are ones that relate to Sigma Xi. So again, the biggest reason a grant proposal is not funded is because somebody reads it and they say, I don't understand what the value of that is. I'm looking at this proposal and I'm saying, you know, even if it's totally successful, I don't see how this is a big advance in our understanding of a broader process or pattern. Now remember, people who are going to be reviewing this in this particular case, they're not going to be necessarily interested in serpentine soils at all. In fact, they may or may not even be interested in adaptation. And they're only, you know, they appreciate speciation as a big question, maybe. That's basically, that's where we're headed from with this, right? So you have to remember, like, big, broad things. People always write grant proposals assuming the reader is just as excited about their topic as they are. That's probably the single biggest reason grant proposals get declined. I had a colleague actually back when I was at LSU who actually he went through a long period where he couldn't get funding. He was working on the nucleolus and the function of the nucleolus. And he was like, but it's so important. I don't understand why it doesn't get funded. But he wasn't able, I think, in his proposals to really convey how amazing the stuff he was proposing to do was and how influential it was and why this is really important. Because, that's one structure. Well, the nucleolus, okay, well, why not the Golgi? Why not, the endoplasmic reticulum? There's a lot of cell structures there. So you have to really sell this. So the significance is probably number one. Methods and approach, how is this going to actually answer the question, right? I said all this last time, but I just want to reiterate that. And tying it back to the literature, has this already been done? Right? If it hasn't already been done, what has been done and how does what you propose to do fit in with what has been done? Now for sigma xi, you have these extra facets. All right? How independent is it of your advisor's research? If you write a sigma xi grant proposal to basically do aim 2 of your advisor's grant, it's not going to get funded. And for good reason, because your advisor's grant has the money for that already. So it has to be something where it's very clearly that you're not doing exactly what your advisor does, but you're trying to do something above and beyond, and that's why you need your own money, rather than just tapping into your advisor's money. And letters have to not say that you suck, or something like that, right? Which you don't, so that's probably a given. It's actually extraordinarily rare, by the way, for sigma xi that a negative letter comes in, and usually when it does come in, it usually reflects more the advisor rather than the student. Okay? All right, let's dive in here. Got the eraser this time. So adaptation to serpentine soils and how it affects speciation. So what is the significance of this? The significance of this probably comes down to this one word. This is the big question for the overall thing. Does everybody know speciation? Speciation is the process of formation of new species, right? Just what it sounds like, right? So speciation is something that, so you start off with your sort of, actually, do you guys have this form from last time? Good, most of you do. Take a second, pull it out if you don't have it with you. This is the Sigma Xi Grant and Aid of Research formula sheet that I sent you guys. Fine, guys. This is not part of the big packet, by the way. This is a bonus sheet. All right. So the first thing you want to do in the first paragraph is set up the research question, right? Why is this something that's really interesting? So you start with a general statement of what is known in this area and its broader significance, right? So speciation is what? It's the process by which, I should say ahead of time, you can vouch for me on this, that we in no way corresponded about this ahead of time, right?
Right.
Okay. He's vouching for me, so this is totally on the fly. So if I falter a little bit, you have to give me a little bit of credit for doing this on the spot in 30 minutes. By which diversity on the planet is formed. Right? That is true, right? If we didn't have speciation, we had a single origin of life way back when, we'd all be the same species, so there wouldn't be any diversity on the planet. So you can say speciation is the process by which diversity on the planet is formed. This is setting up the known thing and the broader significance. You can elaborate if necessary, as it says here. However, alternate interpretations / statement of something unknown. However, somebody tell me, what do you think the however is going to be? What is it we don't necessarily know related to what's written over there?
The specific mechanisms of species for a specific context or environment.
There we go. However, the specific, I don't like the word context.
Mechanisms?
Mechanism, yeah, okay. Well, I'll take that for right now.
Factors, maybe?
What's that?
Is there factors?
Factors is good. Let's use that. Where's that eraser? Specific factors which drive this process are not known, and particularly the role of adaptation to a particular environment, right?
We could frame it in terms of allopathic and sympathetic speciation.
You could frame it in the context of that. You could frame it in the context. Let's stick with one for right now, because there's definitely a different tax for doing it, but that would definitely be a very valid way of also doing it. Particularly the role of adaptation to different environments. Now this is a case where probably right here you're going to need some citations. We'll just do numerical citations. We won't worry right now about what those are. We can fill that in later. I assume that you would easily be able to find a lot of references which say this. There are like whole books that say this, right? Go ahead. Particularly the role of adaptation to different environments. Now, next thing on here says emphasize the feasibility of the hypothesis. That doesn't exactly apply in this case. I mean, this is something that's very clearly feasible. But maybe just emphasize something about this in the context of, the world is very diverse. And you wouldn't put exactly like this. The world is very diverse, so there's many opportunities. Something along these lines. I don't have to fill in that exact section there, but something about along those lines. So, next part. Describe why ignoring this unknown is a problem and greatly diminishing broader understanding. Any volunteers? Why does it matter that we don't know how speciation happens? Why does anybody bother to study speciation?
Well, you come at it from a conservation context that
Absolutely.
understanding diversity and the manufacturing and maintaining diversity is important for conservation.
Absolutely. Understanding the processes that generate diversity. Actually, I'll put generate or maintain. That gives you a little bit more of the conservation standpoint on it. It's essential given human-mediated impacts on the environment or something like that. On the environment and concomitant species loss. Guess what? It is now 12:08. We've completed probably the hardest part of the grant proposal and 1/4 of the total length. It's pretty good. Let's go to paragraph 2. Grab that eraser. We need this? I guess I need to remember the low nutrient part and stuff like that. So this is the end of paragraph 1. Paragraph 2. The advanced possible here. Introduce your system as appropriate with a clear first sentence and elaborate why it is even better than previously used systems. All right? I don't break it up any more than that. So introduce your system as appropriate with a clear first sentence and elaborate why it's better. So let's call it monkey flower, is that right? Yeah. The monkey flower, Mimulus guttatus. Anybody here who doesn't know that? John Willis, who's here in the biology department, works on Mimulus guttatus. Monkey flower, Mimulus guttatus, inhabits a wide range of environments, right? Again, why is appropriate. Including some that are particularly stressful in different ways. I mean, you could focus this. I mean, we just said serpentine soil in this case, but we could focus on particular subsets of this. Including some that are stressful in many plant species. How else can we elaborate this? That's just our topic sentence here. We're not done with this paragraph necessarily. Probably the best thing to do would be elaborate some of these particular environments, right? Maybe say coastal environments, or coastal, not environments, coastal, Mimulus guttatus, are stressed by exposure to high salts, I assume, right? Probably are. High salinity, whereas inland ones. Whereas inland populations have limited water. I don't know. What do they have? I don't know. This is the kind of thing where you actually need to know the system before you write the grant proposal. That usually helps a little bit. I'll just say limited wonder. I don't know. Now let's come back to the serpentine thing. So you said, who was it that said what a serpentine was? Yeah. So tell me more about serpentines. Serpentine soil.
It's found in isolated patches among like normal the normal potatoes environment.
Yeah.
And people think that it might also have low water.
Okay.
It's possible that there's like drought there.
Okay. That's good, that's good. So is there particular toxins there?
There's nickel and there's chromium. Oh, I have a list actually.
Oh, that's okay, that's good, that's good. Hi, nickel and chromium, that's good. That's perfect. So, we have to tie this back to feasibility. We just said now that there's all these different environments. What we haven't actually addressed is this idea of how that could potentially tie into speciation. So maybe let's say, you got making this up out of my butt here, but adaptations observed within a species okay. Try to make this one. I actually finished in under 30 minutes. Adaptations observed within this species affect floral morphology, right? Probably do. How's pollination? Is it wind pollinated or what is it? Bee pollinated? Okay, we'll go with that. Good. Affect floral morphology. Adaptations observed within the species affect floral morphology and can make it get different kinds of bees. But say that in like science speak.
Influence pollinator activity.
Influence. It's not pollinator activity, but you want to say it's getting different pollinators.
Pollinator diversity.
What's that? Pollinator diversity. Very good. Thank you. Very good. Good thing I have a bunch of plant people here. I think. Sounds reasonable, right? You can imagine that maybe, having like, this really limited water supply makes you have this, completely different shape or color of flowers, which makes it not get these kinds of bees, it makes it get these other kinds of bees. If you get the different kinds of bees, then what do you get? You potentially are not interbreeding with the ones that have the other bees, right? You're creating two different populations. So this is starting to sound possible. Right? That's good. You can elaborate that further. I'll leave it at that for right now, but theoretically you could go a little bit further. Paragraph 3. All right, the proposed work. Say what you'll do. It's okay for it to be a pilot project. Be sure to include the test, statistical or otherwise, not just the data collected. That's a common thing actually in Sigma Xi grants that the reviewers get. I mean, it's kind of weird because it's 500 words, right? People always say, well, it's not clear how they're going to analyze the data. I was like, well, it's like group A, group B, and size difference. Like, I'm going to do an ANOVA, whatever. But sometimes they get funky about this. So actually write that in the proposal, okay? I don't know. They're weird. So proposed work. I propose, not we, Sigma Xi grant. You, I proposed, not we. If you write we, they're immediately like, uh-oh, the advisor's doing it, uh-oh. I propose to, what are we going to do? How are we going to test this hypothesis that we've tossed out here?
Measure levels of pre-zygotic isolation between adjacent populations.
Okay, that's one part of it. Let me make some notes here. So basically, how much interbreeding among adjoining populations?
Test the reproductive fitness of artificial classes.
Test the reproductive fitness of what?
Artificial classes.
Ah, there we go. Okay, so this would basically be the pre, and this would be the post zygotic, right? So fitness of I'll put hybrids in quotes. Okay. How are we going to test number 1 here? Well, probably what we're going to do is we're probably not going to actually sit there and follow the bees, right? We're probably going to be doing something where we do something where we look at some molecular marker and just see that, let's say for example you have this patches that look like this. So here's some of the serpentine ones, and here are the, what's the opposite of serpentine? Non-serpentine, there we go. And here's the non-serpentines. There we go. Oh, we see something, I made those purposely different shape, but just so you can identify. And just see that differentiation among these is lower than the differentiation between these. So let's say, for example, you might have populations that are further away that are the same type, but they actually have more interbreeding than ones that are closer of the other type. Does that make sense? That would suggest that they're interbreeding within their own types, but not between types. Okay? I propose to use microsatellites. I don't know, you can use whatever you want. I don't even know if that's spelled right. Do spell check, by the way. Or it doesn't do the little underline thing. I propose to use microsatellites to examine levels of, well, actually, let's just examine, not say levels of, let's say, genetic differentiation among amputatus populations to infer gene flow. So this is sort of the topic sentence, right? We'll come back to the hybrid part in just a second. So in California, there are many neighboring populations of both types. Well, I shouldn't say, I haven't identified the two types yet. Back up a little bit. Neighboring populations, some of which are serpentine, and others non-serpentine. Maybe there's another word for that, but I don't know it. Right? I will use, how many? 12. Sound good? 12. 12 microsatellites. What's the first thing you think somebody's going to say here? What was that? Remember the criteria that were up there for? What's somebody going to say if I just went on from there? Yes, there's going to be a question about feasibility. Can you get 12 microsatellites? Maybe there aren't 12 microsatellites in those. This is the evil reviewer thing. Put parentheses, primers already obtained. From existing genome sequence, because there is already a genome sequence. And, basically measure FST or whatever your favorite measure is of genetic differentiation. Something like that. I predict, it's very clear, that neighboring populations of different type will exhibit higher FST than distant populations of the same type. Is that pretty clear? Everybody understands the test that's being conducted here, right? Any criticisms? Actually, over here, when you have the primers already obtained for existing genome sequence, maybe you probably need like reference 4 or 5. Just to prove that's true. Yeah, Emily.
Going to a non-specialist, I mean, like, how specific do you need to be? Like, measure population, whatever, through FST, or like, do you need to tell them what you're?
You probably don't. I mean, the thing is, so this is a little bit specific for Sigma Xi. They're going to get a lot of proposals kind of like this. So the odds are they're going to have a general idea. They may not know exactly what microsatellites are, but they'll be like, it's some sort of molecular marker, right? They may not know, like, that it mutates using stepwise mutation model. They won't know that kind of stuff. FST, it might be a good idea, since we haven't actually said it, you might be able to say a measure of differentiation. But again, odds are, even if it's not, a straight evolutionary biologist, there's a decent chance they will at least seen that in 4 other proposals that are in this batch. Good question.
What if yours is the first and you're screwed?
Nah, I think you're still good. But if you add in this, a measure of differentiation, I think you're okay. What they may want to, and you can put in there stuff about specific sample sizes. We said here how many markers, but how many sample sizes? You know what your sample sizes are like. How many are you going to get from each population? You know how many populations. So these are the kinds of details you'll want to fill into this as well. So I'm not going to keep doing that right there because I'll take too long. Any questions on that so far? All right, let's come back to the other part. I'm actually merging paragraphs 3 and 4 a little bit in this one. The way it says here, it says to do the first one to be the test and stuff like that, and then number 4 to be the predictions. But I'm actually going to, in this case, since we actually have sort of 2 different projects that are using the same individuals, I'm actually kind of separating those out and putting the predictions next to each one. Because you don't want to have all this stuff and then all the predictions. That would just get confusing. Okay, so yeah, keep that in mind. This isn't like dogma. This is just one way of doing it. Got 6 minutes to wrap up this grant proposal. All right. Additionally, I will artificially, not inseminate, what do you say? Artificially cross or pollinate, whatever. Cross Minuscutatus from population 1 and population 2. I mean, I'm abbreviating here now, but you guys can see the general idea. Basically, we're going to artificially pollinate some of the serpentine to the non-serpentine and measure fitness. How are you going to measure fitness?
Seed set.
Seed set. Very good. Better than, don't even say fitness. Just say, I'll measure seed set. Maybe, what's it, you know, we always say for Drosophila, say egg to adult viability, I assume. Seed to plant, I don't know.
Seed to flower.
Seed to flower, there you go. Etc. And compare to there. And compare them to the various controls. I predict what? What do you predict? If there is some hybrid problem here, what do you predict?
The population 1, population 2 cross will be worse.
Exactly. The seed set will be lower in this group than in this or in this. So you just write that up. Finally, last thing you want to do. So we've already stated our predictions. What now we want to do is we want to say how this will stimulate further work and relate it back to the big picture. So this is our wrap up. This is actually very important. A lot of people stop the proposal here. And people are reading and they're like, I predict blah, blah, blah. Okay, wait, what were we trying to do again? It's a very common thing in proposals not to bring it back to the big picture. So at the very end, you come back to the stuff we were talking about at the very beginning. Something along the lines of speciation is poorly understood, or the mechanisms of speciation are poorly understood, right? But knowing the role of adaptation to different environments well, what? Why is it important? Going back to the big picture. Why do we care?
Will shed light on mechanisms of speciation.
That's a little too circular. That is true. That's a little too circular.
Increase our understanding of how environmental change affects diversity.
Beautiful. Environmental change will affect our planet. You can add to it something like, we always, researchers tend to focus on specifically the loss of species. However, studying this will give us some idea about the loss of species that basically we never get given a chance to form by losing environments. Something along those lines, right? Sounds good? There we go. Right at 12:25. 30-minute grant proposal. This is a topic that, like, you know, I know a little bit about, I mean, because I actually do work in speciation more generally, but I definitely don't work in mimulus. You guys can do this. Just follow this 4-paragraph formula. Think about whatever it is you're working on, and you should be able to apply this fairly quickly and straightforwardly to, you know, hammer out something on your topic. Now you're gonna have to do a lot of reading, because I was just making up stuff to fill in the gaps here. Obviously, don't do that. So, you know, fill in, like, you know, what some of the research has been done, how it relates to other things. I didn't relate it very much in the first paragraph to other research, it's probably as much as I should have. But that's the kind of thing you'd want to fill in that actually wasn't done here. Are there any questions on that? Yes?
So the person to write our letter would be our, like, if we're in a rotation, our
Yes.
advisor now?
Yes.
And do we?
You need 2 letters. So you need one from, presumably, like, I'm assuming you'd be writing it in the context of the lab that you're in right now, too. So if that's the case, then yes, one of them should be from your current research supervisor who's doing your rotation. The other letter should be somebody who you've done research with in the past. So let's say, for example, you did master's research or undergrad research, have that person write the second letter. If you haven't done research in the past, which is true for a small subset here, then just somebody who, you know, interacted with a lot and got excited about research with. So what was the other follow-up to the question?
So do you explain to them that this may not be the project that it's gonna end up with?
Yes,
Or?
yes. You can tell them that, you know, you say, I'm writing this now, in the event that I end up in this lab, there is a possibility I may end up in another lab, in which case, you know, that's okay. So I tell them that. However, it would be very nice, and this is something to really urge your letter writers to do, is urge them to say something about the project. A lot of people, what they'll do, especially like, say, your past research supervisor, they just like copy, paste, you know, their old letter. I mean, I do this actually. It's a good idea to ask them to say something about this project, so they actually know that, you know, when the reviewer's getting it, that you've actually discussed this project with somebody besides just your research supervisor. They don't have to, like, you know, go on for pages about it, but acknowledging something, you know, even like 3 sentences saying, I think her present proposed research on, you know, adaptation to serpentine soils and Humulus cutatus sounds like it'll be very promising, something like that. Certainly your current research supervisor should emphasize that quite a bit. Now this actually leads to another good point that I didn't bring up yet. You have a 500-word limit, right? What you can do is, let's say, for example, there's a point you wanted to bring up and you really couldn't fit it anywhere in here. There's a cheat that you can do. You tell your advisor, hey, you're writing a letter too, right? Maybe you could slip in this particular point. And the advisor can actually get away with doing stuff that you can't. Because, like, with your stuff, you have to have all the citations and things like that. They can just say, this is the best research ever, which you can't say that. I mean, that would look really weird in your proposal. This is the best project ever. But the advisor can totally do that. So they can both sell your project, you know, help you in that context, and also they can fill in any little particular things. Let's say, for example, there's one study you think a lot of people might know about but it really isn't as good. They can just say in their letter, this is going to be much better than the study by so-and-so. And that's totally fair. Is that a good little cheat? Yeah?
How far back, like, if you're writing one, say, in your 4th year or your 3rd year or something, do you still need to get someone who you previously worked with?
No, at that point, if you're in your 3rd or 4th year, you can get like somebody on your committee or something like that. That's totally fine. Yeah, good question. Sorry about that. I should have brought that up initially. I forgot that some of you are at like 3rd years in here and stuff.
And your committee will, like, know you and your research well enough to recommend you, even if you're not in their labs and stuff?
Assuming that, like, you didn't just pick your committee, there's a decent, like, let's say, for example, especially if you're post-prelim, they should definitely know you. If you're pre-prelim, assuming you've had a couple of committee meetings and talked about research and things like that, then it should probably be fine. I mean, do whatever you're most comfortable with. I mean, if you feel like, you know, I really think in my case, my committee doesn't know me, but my undergrad advisor does, go to the undergrad advisor, it's fine. There's no requirement here. The only thing you really absolutely want to have is one from your current lab. That's the absolute. The second one is just basically like somebody who's research-oriented. That's all you really need. Yeah?
What if your proposal doesn't really have anything to do with the current lab you're in? It's just like a side project that you're interested in that's feasible?
Mm-hmm.
Do you, like, if
Is it something that you think you would do in that lab, or is it something you wouldn't even think you would do in that lab?
You mean, like, I mean, if it's like this, like you could go collect this data and sort of just do it yourself. I mean, like you could actually do it in the lab, but it wouldn't be associated with the research that's done normally in that lab.
But let's say, for example, in your case, let's say that, is this a project that you were assuming that you would do it, you would be more likely to do it with your present supervisor?
No, yes.
Oh, okay. In that case,
should you go with, like, talk to some other professor that has more experience with this type of stuff?
that's a tougher question. That doesn't usually come up. So in this case, this is the question that basically, let's say, for example, like you're in your 1st rotation, but this is a project that would be relevant to your 2nd rotation, and you haven't actually worked with this person at all yet. I probably would not recommend you get the letter from the second one, because that person has zero experience with you right now. At that point, it's probably better to go ahead and get with the first one. It's a little bit funny though in that situation. It's probably, I mean, if you can, it's probably good to gear it for your current lab, but if not, you know, give it a shot and see what happens. Worst case scenario, again, 500 words, 30 minutes. Yeah?
So just sort of generally about grants, I was looking at the NSF grant, and they have all the categories.
Yes.
But what if you're doing like evolutionary genetics? How do you pick which category to put yourself in?
It doesn't matter. I mean, let's say for sigma xi, you just pick evolution, something like that. You know, basically just figure which one's a little bit heavier, but if you're miscategorized, it's not a big deal. At places like NSF, they will potentially reassign where your grant is going to go. So let's say, for example, you say, I think this goes into evolutionary mechanisms. They're like, no, this totally goes in evolution, in animal behavior, for example, or something like that. They will actually just shunt it over there, or they'll have it jointly reviewed, things like that. The sigma xi doesn't freaking matter. It's just like a box that somebody picked up. It's totally fine. Yeah.
So in general, for asking people to write you letters of recommendation, does it matter what their title is? Like, if you have two people that you might want to ask, should you ask the person who's higher up?
PhD is probably essential. Like,
Okay,
I wouldn't ask like, you know, the grad
assistant
student you
professor
work
versus
with.
full professor versus no,
No,
it doesn't matter.
I don't think it matters at all. Yeah.
I was just wondering, like, for this specific grant, how much you should go into, like, the instruments that are available to you?
So I didn't come back to that part yet. So this is just the body of the proposal. Now, you actually still have to have a budget. So like, it's unclear, for example, from this, what exactly you'd be asking for. Since you're doing microsatellites, odds are your budget, and you have a budget and a budget justification for pretty much any grant proposal. Your budget, in this case, may be something like, you know, $250 for polymerase. You said you already had the primers, you can't take that back now. It's not locked in there. But maybe a big part of it is, you know, 400 airfare to California. And then 350, actually let's make it 150 gas, 200 lodging. So basically, you're going to all these different places and saying what you got. Now, you don't actually have to say it. Like, I mean, it's assumed that if you're using something like, let's say you say you're going to use microsites, it's assumed you have a thermal cycler. It's assumed you have pipetters. If there's some really unusual piece of equipment, let's say, for example, you're using, I don't know, maybe let's say, for example, you need high access to a mass spec or something like that, something which just isn't necessarily like in every lab around there. It might be a good idea to have your advisor just mention in the letter, all resources, including the mass spec, are available in our lab. You probably don't want to squeeze it into your proposal. You may be able to, if necessary, you can just add it really quick to the budget justification. But I wouldn't use up your 500 words talking about those kinds of things. Yeah?